Okay, let's start chapter 3. Thank you for your patience this morning. Okay, test. There we go. We have some sound now. We're starting chapter 3 this morning. Please, if at any time you have questions, raise your hand. I'll repeat the question so we make sure we get it on the recording. If there's any time that I don't make sense, please let me know. Again, this chapter is very, very important. Okay, we're talking about organic molecules this morning. So let's just right off the bat get a definition of what exactly is organic. Okay, organic is anything that is living or has lived in the past. The word organic means living or has lived in the past. Of course, the opposite of that, inorganic, would be non-living. Okay. We talked in chapter 1 and 2 about how all things are made of the same chemicals. Well, all living things are made of the same elements, basically. Does anybody remember what those 6 elements were that make up all living things? Hydrogen, carbon, oxygen, nitrogen, sulfur, and phosphorus. You see them sitting right there on the screen. Yes, those 6 elements make up 98% of living mass. Okay? So in this chapter, we're going to talk about how those 6 elements come together in molecules to make up your body or your mass. Okay? How they come together in molecules that we're going to call carbohydrates, lipids, proteins, and nucleic acids. So the entire lecture today is going to be on these molecules, how they're made, and characteristics of each category. Okay? First of all, before we get started, let's just get some general characteristics of what an organic molecule is and how it compares to an inorganic molecule. Okay? Inorganic molecules have primarily ionic bonding, and organic molecules have covalent bonding. We talked on Thursday of last week, Tuesday of this week, whatever day it was we met last for lecture. We talked about ionic and covalent bonding, and we said that ionic bonding was when electrons are transferred. That creates charged atoms called ions that are attracted to each other. Covalent bonding is when atoms share electrons. Okay? Inorganic molecules obviously contain ions, and organic molecules always contain hydrogen and carbon. Inorganic molecules contain ions, and organic molecules contain hydrogen and carbon. Carbon especially. I want you to associate organic molecules with carbon. Okay? So if I give you a list of molecules on the exam and say pick out the ones that are organic, what are you going to be looking for? Hydrogen and carbon. Okay? So tell me right now, is water organic? It has to have carbon. Okay, it has to have carbon. So water's not an organic molecule. Water, we learned in chapter 2, is essential to life. All living things have to have water, but it's not an organic molecule. Water's not alive. Okay? Inorganic molecules have a small number of atoms. Organic molecules are very, very large, lots of atoms. As a matter of fact, in a few moments, we're going to call these as a group macromolecules. And macro, we learned in our root words, means large. Okay, so these are large molecules. And of course, inorganic molecules are found in things that were not living or non-living, and organic molecules are found in things that are living. Okay, so we said all organic molecules have carbon, right? So let's take a look. Let's just talk for a moment about carbon as an element. Carbon has an atomic number of 6. What does that mean? That means it has 6 protons. Yes. Well, atoms are electrically neutral in nature, so that also means it has 6 electrons. With 6 electrons, how would those electrons configure themselves? 2 in the first shell and 4 in the second shell. Okay, the second shell is the valence shell for carbon. So how many would it like to have a full outer shell? It needs 4 more. That's right. So it's half full, which is kind of a precarious situation. It gives carbon some characteristics that are really, really important. Okay, has 4 electrons in the outer shell. So it needs 4 more. Carbon loves to form covalent bonds. And we learned in chapter 2 that these are really, really strong bonds. Carbon loves to form covalent bonds. And we also learned that atoms can covalently bond with more than 1 electron being shared. Sometimes you can have 2 electrons being shared or 3, and those would be double covalent bonds and triple covalent bonds. But with 4 electrons in its outer shell, carbon can bond with up to 4 different elements, 4 different atoms. It can even bond with other carbon atoms to form these strong covalent bonds. So what you have with carbon forming covalent bonds with other atoms is you have the ability to form these long chains that we talked about in chapter 2. So you can have carbons bonded with other carbons that are bonded with other carbons, and you end up with these long chains of carbon atoms. Well, these structures form the backbone of these organic molecules that we're going to talk about. We call them hydrocarbon chains. So bonding with other carbon atoms produces long chains that we call hydrocarbon chains. So let's take a look at an illustration of a hydrocarbon chain. Okay? You see there that we have carbons and hydrogens. Each one of these lines indicates that there's being a single electron being shared. So this carbon right here is sharing an electron with another carbon atom. It's sharing an electron with a hydrogen, and another hydrogen. So that carbon atom thinks it has a full valence shell. It's sharing 4 electrons, as are all the other carbon atoms in the illustration, and the hydrogens are sharing electrons, so they think they have a full valence shell. As this structure stands, it's non-polar. There's no polarity. These are all covalent bonds, electrons being shared equally. So there's no polarity associated with a hydrocarbon chain that has no other structures or atoms attached to it. Okay? So if it's non-polar, how do hydrocarbon chains interact with water? It's hydrophobic. Yes, that's exactly right. So hydrocarbon chains without any other atoms attached are hydrophobic. Okay? Now, hydrocarbon chains can come in all sizes. You can have dozens of carbons, even hundreds of carbons all bonded together, or you could just have 1 carbon with 1 hydrogen. Okay, so this is not, it doesn't have to be a certain number of carbons there. So they come in different sizes. Sometimes we see other atoms attached to hydrocarbon chains in these organic molecules. And again, this structure is going to form the backbone of a lot of these organic molecules we talk about. And we see other atoms attached to hydrocarbon chains. Okay. We sometimes see groups of atoms appear together in lots of different places. Okay, there are atoms that like to hang out together. And we'll see them attached to these hydrocarbon chains. We call those groups of atoms functional groups. Okay? And a functional group is just a group of atoms that has a certain pattern. They like to hang out together. And we see them attached to these hydrocarbon chains in these organic molecules. It's a cluster of atoms with a certain pattern. And they like to hang out together. And we see them on these molecules. And this cluster of atoms has properties that it takes with it. It always behaves in a certain way. Whatever the properties of that functional group, it takes those properties with it. And it can actually change the polarity of these hydrocarbon chains, these organic molecules. So a functional group we find attached to carbon chains, And it's the same atoms that hang out together, and they always act the same way. And guys, you already know one of them. We talked about it. We talked about the atoms in chapter 2. Do you guys remember when water dissociates? It dissociates into equal numbers of what? Hydronium and hydroxide. What is hydroxide? What is, he said, 1 oxygen and 1 hydrogen. And what is the charge associated with that? It's a negative, okay? So hydroxide is OH negative, right? Well, we see OHs attached to hydrocarbon chains a lot, and they're always negative, okay? Except when they attach to a hydrocarbon chain or another molecule, instead of calling it a hydroxide, we call it a hydroxyl. And that's the first functional group right there. So you see it and you know it brings a negative with it. So that's an example of a functional group. There are others that we're going to talk about attaching to these hydrocarbon chains. We'll talk about carboxyls. That's an acid group that we find in amino acids and fatty acids. Amine groups contain nitrogen. We find those in proteins. Phosphate groups, we'll talk about that in our nucleic acids and in some lipids. So these are just some functional groups that we're going to see when we talk about these organic molecules. Okay? I'm not going to make you memorize these functional groups. This is not chemistry. I just kind of want you to know what a functional group is and that it really can change the polarity of a molecule. For instance, let's take a look at that. If we have a hydrocarbon chain right here, and we know that this right here is nonpolar, so it's hydrophobic, right? And this is a functional group that's attached to that hydrocarbon chain. We know that OH has a negative associated to it, so it literally changed the polarity of that molecule. We have an area that's nonpolar and an area that's polar on that molecule, okay? So let's make sure we understand, just to kind of summarize what we've talked about so far. Molecules with only carbon and hydrogen are nonpolar, hydrophobic, which means they're not attracted to water, right? We can add these functional groups, and they can make it polar or hydrophilic or attracted to water. So that just kind of summarizes what we've talked about so far. Okay, let's talk about how these different structures can be. Before we get into organic molecules and how they're made and the different characteristics, I want to introduce you to the concept of isomers. We're going to see molecules that are called isomers, which means they have the exact same molecular formula. Now, what is a molecular formula? Let's go back to chapter 2. What is a molecular formula? That tells me the numbers of the different atoms of the elements that it takes to make the molecule. Okay? So I have, as an illustration here, 2 different substances. The one on your left is glucose, and the one on your right is called fructose. They're both sugars, and they both have a molecular formula of C6H12O6. They both have 6 carbons, 12 hydrogens, and 6 oxygens. They have the exact same molecular formula, but they're 2 different substances. What makes them different is that they have a different arrangement of atoms. Okay? Iso, if you remember from your root words, means equal. So these have an equal number of atoms, but they're 2 entirely different substances. Okay? Okay. These carbohydrates, lipids, proteins, and nucleic acids, we're going to call these molecules, these organic molecules, macromolecules because they are huge. They are large molecules, and they are composed of smaller molecules. They are very complex in structure, very complex. So we're going to call them macromolecules. They're large molecules, a large molecule, a polymer, but they're built from smaller molecules that we're going to call monomers. What does poly mean? many. What does mono mean? One. So we're going to call the individual units that make up these large molecules a monomer, and we're going to call the large molecule, we're going to call the protein a polymer of those monomers. Okay? So polymer and monomer are generic words. And the way that you can think of this, a good example would be a freight train. A freight train would be a polymer. It's lots of individual units that are all joined together. So of a freight train, what would be the monomer? The individual cars, okay? So we're going to talk about proteins being a macromolecule. It's a polymer. It's made of lots of individual units, and the individual unit or the monomer for a protein is a amino acid. Okay, so each one of these groups of organic molecules is going to have a monomer. Okay, so we take monomers, these individual units, and we bond them together to form polymers. Okay, so polymer is a long molecule consisting of many building blocks called monomers. And again, these are generic terms. When we get into carbohydrates, we'll talk about what is the monomer for a carbohydrate. When we get into proteins, we'll talk about what is the monomer for a protein. When we get into nucleic acids, what's the monomer for a nucleic acid, okay? Each class of macromolecules has a specific monomer. Again, a great example is protein monomers or amino acids. Okay, let's talk about how to build these molecules before we get into the individual classes of organic molecules. It doesn't matter if we're talking about building a carbohydrate or building a protein or building a nucleic acid. The reaction to build all of those molecules is basically the same, okay? To synthesize a polymer, remember your prefix SYN means to put together. So synthesis is the putting together of a molecule or making a molecule. So if we're making a polymer, that means that we're taking lots of monomers and joining them together. The reaction that does that for all of these molecules is called dehydration or condensation synthesis. Okay, now don't let those words intimidate you. Let's think about what those words mean. What does dehydration mean? If you dehydrate something, what does that mean? You're taking the water out. That's exactly right. Some books even call this condensation synthesis, which is basically giving you the same connotation, that water is coming out. Okay, so synthesis is putting something together. So these words tell you exactly what's happening in this reaction. You're putting something together and water is going to exit. Okay, this is what happens. This is the reaction that occurs when monomers join together. Okay. So let's take a look at this illustration for just a moment. This is a generic illustration. Okay, this could be a nucleic acid, this could be a protein, it could be any of those, okay? This obviously is a polymer right here. This is a monomer, a generic monomer. What I'm trying to say is that for every monomer, for all of these organic molecules, there's going to be a hydrogen on one end and there's going to be an OH on the other end. Okay, so obviously what makes a protein monomer different from a carbohydrate monomer would be what's in that area right there. Okay, so if I'm going to join this monomer to this polymer, okay, I'm going to remove a water molecule. So the OH comes off that monomer, the H comes off that end, they join together and form a water molecule, and where they once existed, we now have a bond formed. Okay? So the bond is forming and water is being removed. Now I have a polymer that is made out of 4 monomers. And notice that when this is bonded, I still have an H there, waiting for another monomer that would have an OH, water would be formed, and another monomer would join. Okay, so this is how we build proteins. This is how we build carbohydrates, etc. So think about this for a moment. What if I wanted to break down an organic molecule? If I've got a polymer, an organic molecule, and I want to break it down, what would I have to break it down? I would have to have water. That's exactly right. So the breakdown of a polymer is called hydrolysis. And again, the word is telling you exactly what's happening. Hydro means water. And you learned this in your words. What does lysis mean? To break, to burst, yes. So this is telling you what's happening in this reaction. The reaction of hydrolysis is literally the breaking of a bond with water, okay? So I'm going to take this polymer right here. I'm going to break it down into the individual monomers by using water. It's the reverse of dehydration synthesis. So the water is going to be used to break that bond. The water splits, the H goes here, the OH goes there, and I've now broken a monomer from the polymer. Okay? Okay, so here in a summary, we have the synthesis and breakdown of polymers. We have the monomers that undergo dehydration synthesis. Water's coming out to join those monomers to make a polymer. To break down a polymer, we would have to have water going in, with the reaction being called hydrolysis. And then you would have that polymer broken down into monomers. So this is how you build proteins and break them down. This is how you build carbohydrates and break them down. And this is how you build nucleic acids and break them down. Have you ever tried to break down lipids, lose weight without water? You can't do it. You have to have water to break down those molecules. Okay? So now let's get into our different categories of organic molecules. Let's start with carbohydrates. This is my personal favorite. I always put it first because I love sugar. Okay? I love sugar. Sometimes we call carbohydrates saccharides even. These are sugars or saccharides. Does anybody remember the first artificial sweetener? What was it called? Saccharin, that's exactly right, saccharin. So these are sugars or saccharides. Carbohydrates as a group of molecules serves two functions. One of them you're very familiar with, and the other one you might not be so familiar with. One of the functions of this group of molecules is energy. Okay, before you go out for the big game, the team goes to Pasquale's or Pizza Hut or somewhere and they carb up, right? Pizza, pasta. That's why I like playing sports, get to carb up. Okay. The other function of carbohydrates as a group is that some of them form structural roles in organisms. That means they're a building material. They become part of that organism's structure. So we're going to talk about those two different groups of carbohydrates. But first of all, let's define what a carbohydrate is so you'll be able to recognize it. Okay? Carbohydrates, by definition, a carbohydrate is a molecule that contains the elements carbon, hydrogen, and oxygen, and the atoms will be in a ratio of 1 to 2 to 1. Okay, what does that mean? That means that in that molecule, you'll only have carbon, hydrogen, and oxygen, and you're going to have twice as many hydrogens as you do carbon and oxygen. So a good example would be those isomers we talked about a while ago, glucose and fructose. Their molecular formulas were C6H12O6. So we have twice as many hydrogens, 12, as we do carbon, there's 6 of those, and oxygen, there's 6 of those. So if we had C3H6O3, that would be a carbohydrate. Okay, so if I give you a list of molecules and say pick out the carbohydrates, what are you looking for? Carbon, hydrogen, and oxygen in a ratio of 1 to 2 to 1. Sometimes when you get into the very, very large carbohydrates, that ratio might be off by an atom or two, but very, very close. Okay, the monomer of a carbohydrate is called a monosaccharide. Mono means 1. So this is literally one sugar. This is the simplest form of a sugar. These are the monomers of carbohydrates. And these simple sugars, these monosaccharides, can be used as energy themselves. They can be used as energy themselves, as fuel. And we use glucose as fuel, okay? Or they can be bonded together and combined into a polymer for storage of energy. Okay, so we run into glucose by itself, and then we have lots of molecules that are glucoses bonded together. A simple sugar, a monosaccharide, will always have between 3 and 7 carbon atoms. That means it's a monosaccharide. If you have a carbohydrate that has more than 3 to 7 carbon atoms, it's not going to be a monosaccharide. So our example of glucose and fructose, what was that molecular formula? C6H12O6. So they both have 6 carbons. Does that mean that glucose is a monosaccharide? Yes, glucose is a monosaccharide. Glucose and fructose both have 6 carbons. Other examples of monosaccharides that we're going to talk about are a sugar in particular named deoxyribose and ribose. These are sugars. We find those in DNA and RNA. Both of those sugars have 5 carbons. And go ahead and write down in your notes beside that, a 5-carbon sugar is called generically a pentose sugar, Pentose. Do you know what a pentagon is? It has how many sides? 5. That's exactly right. So 5-carbon sugars are called pentose sugars. And all of these would be monosaccharides, okay? When we look at the structure of a monosaccharide, we often look at them linearly, like you see here in the illustration. This is showing you the arrangement of the atoms in these different sugars. These are triose sugars. They have 3 carbons. These are pentose sugars. There's ribose and there's ribulose. These are 5-carbon sugars. These are hectose sugars. That means they have 6 carbons. So there's glucose, there's fructose, there's galactose. Those are all 6-carbon sugars. So we can look at them linearly, or sometimes when we are looking at illustrations of molecules, they show the sugar as it would be structured in an aqueous solution. What does that mean? In water. That's exactly right. And you're how much water? 70 to 90% water, right? So we see these sugars form rings in aqueous solutions. Don't let that confuse you, okay? What happens when these sugars are placed into aqueous solutions, take a look at fructose right here, or glucose, either one of those. What happens is this carbon will come around and bond with that carbon right there. And it forms a ring, literally. And it's going to look like this, okay? So here you see fructose right there. That's your fructose linear structure, and that is your fructose ring structure. And the reason I introduce this, I'm not expecting you to know that where the atoms are or anything, but I want you to realize that that hexagon right there that you see is illustrating fructose. That's a 6-carbon sugar. Because when we look at these other molecules, you're going to see this illustrated as a hexagon. And I want you to recognize that that's a sugar, okay? And I don't want you to get confused that what is this and what is this and how are they different. They're the same thing. It's just that in water, this structure loops around and forms a ring. Okay? Okay. Okay, when you bond 2 monosaccharides together, when they are bonded together, it is called a disaccharide. Di means what? 2. That's exactly right. It's a disaccharide. Quick quiz. What reaction would be used to bond two monosaccharides together? Dehydration synthesis. Very good. Excellent. Okay. So some disaccharides you may be familiar with. Maltose is a disaccharide you may have heard of. You find it in our digestive tract. It's actually glucose bonded to a glucose. Okay. Sucrose is a plant sugar. That's a glucose bonded to a fructose. Real sugar. And lactose is milk sugar, and that is a galactose bonded to a glucose. So all of these are disaccharides. And if you'll notice in the illustration, they are using these different symbols there, and you see that maltose is glucose and glucose, sucrose is glucose and fructose, etc. Some of you lack the enzyme necessary in your digestive system to break that bond right there between the glucose and the galactose. What would you be called? Lactose intolerant. That's right. Okay. Okay. When you have lots of monosaccharides bonded together, we call it a polysaccharide. Poly means many. Okay. Of these polysaccharides, there are some of these polysaccharides that are used for energy, but there's also a whole group of them that's used to make the structure of some organisms. Some organisms produce these polysaccharides as part of their structure. Let's take the energy ones first and talk about those. Let's talk about the polysaccharides. Again, a polysaccharide is lots of monosaccharides bonded together, and it's used for energy. The first one's called starch. Starch is literally a polymer of glucose molecules. It's glucose bonded to glucose bonded to glucose, and this is what it looks like. Notice the hexagon there. So that would be a glucose, that's a glucose, that's a glucose, etc. Starch is glucose, 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 bonded together, lots of them. Well, name me some things that contain starch. What is starch? What? Potatoes. I love potatoes. What's another one? Pasta is another one of my favorites. My mouth is starting to water. What's another starch? Bread, yes, and rice, all the things I love. So tell me, what are all those things? Pasta, bread, potatoes, what do they all have in common? They're all carbohydrates and fattening. Yes, they are. You're exactly right. But where do they come from? From where do they come? Plants. That's exactly right. So we get starch from plants. Guys, this makes so much sense. Biology makes so much sense. That's why I just love it. We said in the last lecture that plants that are autotrophs, right? They're autotrophs. So what are they doing? They're taking energy from the sun and they're storing it for us. Well, we learned in chapter 2, how do we store energy? In living systems, how do we store energy? We store it in the bonds between atoms, right? When atoms bond together, the energy is stored. So the plants are taking the sun's energy and they're building a molecule for us, using that energy and storing it. And the molecule they build is a monosaccharide. It's a glucose. So plants are building glucose. Well, plants aren't going to use all that glucose at one time. So what they do is they use it, but then they've got extra, so they bond the glucoses together literally to store them and save them for a rainy day when there's no sunshine. Okay, so they're putting glucoses together, saving them so that when they need energy, they'll take that starch, that is glucose bonded together, break it down and use the individual glucoses for energy. So starch is literally how plants store energy. Doesn't that make sense? Okay, so there's another energy polysaccharide called glycogen. It's also a polymer of glucose. Lots of glucoses bonded together. Glycogen is to animals as starch is to plants. Okay? So this is how animals store energy. And again, this is glucose bonded to glucose, bonded to glucose. So it's a polymer of glucose. So what are we doing as an animal? We're taking glucose molecules and we're bonding them together. We're going to store it in the form of a molecule we call glycogen. It's stored in your liver. Okay, and again, this is for temporary energy storage. This is not your long-term storage of energy. That would be a lipid. Okay. This is temporary. So what you're doing is you're taking glucose from the foods that you eat and you're bonding them together. You're going to store it in your liver as glycogen. So let's say I get long-winded today and I start preaching and we don't get out of here till after lunch. So what's going to happen to your blood sugar between now and then? It's going to drop. That's exactly right. So in that case, your liver is going to take this molecule we call glycogen and start breaking it down into the individual glucoses and release that glucose into your bloodstream. Okay? So it's stored in the liver and it's released as glucose to keep your blood sugar normal. And I promise I'm not going to preach till lunch. The other group of polysaccharides that we said were for structural purposes includes two molecules that you may be familiar with. One of them is called cellulose. Cellulose is also a polymer of glucose. It's very complex, though. The bonds between the glucose molecules are very complex. But it is glucose bonded to glucose bonded to glucose. We find cellulose in plant cell walls. It's the major component of the walls that enclose plant cells. Some cells have additional coverings beyond the cell membrane or the plasma membrane. Animal cells do not. Okay, plant cells do. Plant cells have what's called a cell wall that is there for structure. It gives rigidity to the cell. It's for protection, and it's made out of cellulose. Okay, so there's glucose, glucose, glucose. This cellulose is very important in our diet. Very important. When you eat plants, if you have a salad for lunch and you start chewing those plant cells, you're going to destroy the cells, you're going to bust them open, you're going to macerate them, and you're going to break down the cell. Well, you break down all the cell and it goes into your digestive system. All of that cell is used for energy and for nutrition except for the cellulose. When you eat plant cells, the cellulose that makes up the cell wall of those plant cells goes straight through your digestive system undigested. Of all of those atoms there, you are not able to obtain any energy from cellulose, nor can you obtain any nutrition from cellulose. It goes through you undigested, but yet it's extremely important in our diet. What do we call it? Fiber, yes. What does it do? Because it goes through your system undigested, it helps to keep you regular. Very important, okay? Very difficult molecule to break down. Not only can you not break it down, but I'm sure that you're familiar with herbivores like cows and sheep, etc., that only eat plant matter. Did you know that they can't even break down the molecule that we call cellulose? They actually have a bacteria that lives in their gut that does that for them. Okay, so even they can't break it down. They have bacteria that aid in the digestion of cellulose. Another structural polysaccharide that you may be familiar with is called chitin. Chitin, again, forms the structure of some organisms. It is a polymer of glucose. It's used in the exoskeleton of insects, crustaceans, the exoskeleton of things like roly-polies are crustaceans. They're crunchy. If you eat roly-polies, they're crunchy. If you eat insects, they're crunchy. This is very hard to digest. It can't be broken down. This is why you don't eat the peeling on your shrimp. You couldn't digest it if you could. Also, fungal cells have a cell wall, just like plant cells have a cell wall. But fungal cell walls are made out of chitin instead of cellulose. Okay? Cellulose makes up plant cell walls. Chitin makes up fungal cell walls. Okay, well, let's go on to lipids now. Lipids are our second group of organic molecules. Very diverse group of molecules. They are found in every type of plant and animal cell, and they serve lots and lots of different functions. They make up the cell membrane. The hormones in your body are lipids. They are energy storage molecules. Some lipids are for protection. Our nerves are insulated by lipids, and they also serve to waterproof and are a factor in buoyancy. Very diverse group of molecules. They are hydrophobic. What does that mean? They fear water. They are repelled by water. As far as the definition of what a lipid is, that's a molecule that contains carbon, hydrogen, and oxygen. It's going to have less oxygens than carbs. It's not going to be that 1:2 to 1 ratio. So if you have to pick them out, they wouldn't have that ratio. Lipids are unique as an organic molecule in that they don't polymerize. This is an exception to the polymer-monomer example. Okay? To make a lipid, you do have unit molecules, but you're not going to bond them together in chains. You do have two monomers that come together, but they don't form a chain. Okay? And we'll talk more about that when we talk about how to make a lipid. But there are 4 groups of lipids we're going to talk about today. We're going to talk about fats and oils. We're going to talk about phospholipids, steroids, and waxes. We're going to spend most of our time today talking about fats and oils and phospholipids because those are the ones that are more relevant to this course. If you're taking anatomy, next semester you'll learn a whole lot about steroids and waxes. We'll tell you what they are. We'll talk about the structure, but we're not going to spend a lot of time on them. Let's start with fats and oils. Fats and oils are primarily for long-term storage of energy. Fats and oils are composed of 2 different unit molecules that come together to form the molecule of fat or the molecule of oil. To make a molecule of fat or oil, you need a glycerol, which is highlighted here in green, and 3 fatty acids. So to make a molecule of fat or oil, you're going to have a glycerol and 3 fatty acids. Notice that the fatty acids are hydrocarbon chains. Look at that. Those are hydrocarbon chains. And notice that most of them, these 2 right here, have single bonds, but look at that one right there. What are those 2 lines telling me? It's a double bond. That's exactly right. So that's a double bond. Those 2 carbons are sharing 2 electrons. But those are hydrocarbon chains. And to put a molecule of fat together, what's the reaction? Not hydrolysis. Okay, you're in the right area of the lecture. Condensation synthesis, because look at what's highlighted here. The O and the H right here are highlighted on the fatty acids, and the H from the glycerol. They come together, water comes out of the reaction, and you would have a molecule of fat. So you would end up with 3 water molecules coming out of that reaction, that condensation synthesis reaction. And what you would have formed when those bond together is called a triglyceride. That would be a molecule of fat or oil. That's why we call them triglycerides. It takes 3 fatty acids per glycerol to make a molecule of fat or oil. And you have 3 waters coming out of that condensation reaction. Okay? So, what we said, fat or oil. What's the difference? If this is a molecule of fat or oil, how do I know it's a fat or an oil? Well, how do I know the difference? Does anybody cook in here? Do you cook? Go to your pantry and pull out an oil for me. What would you pull out? Olive oil. Very good. Anybody else want to add to it? What else would you pull out? Vegetable oil. Very good. Yes. Okay, now, who else cooks in here? Okay, go to your kitchen and pull out fat for me. What would you pull out? Lard. Oh my gosh, that's my favorite. Lard or butter. That's exactly right. Very good. And both of those are wonderful. This lecture makes me so hungry. So tell me, class, what's the basic difference between those 2 groups of substances? One's a solid and one's a liquid. That's exactly right. So let's talk about now molecularly what makes the solid a solid and what makes the liquid a liquid, because both groups are triglycerides. It's a glycerol with 3 fatty acids. Okay? We call those 2 groups saturated and non-saturated fats. Okay? So let's take the non-saturated or unsaturated first. Those would be your oils. Okay, you said corn oil, olive oil, vegetable oil, the ones that are liquids, okay? Notice right here in the example that this would be an example of a fatty acid found on an unsaturated fat, an oil, okay? Notice that there are double bonds right there between the carbon atoms. Okay? Double bond means that the carbon can't have another hydrogen right there. If that carbon has a double bond with that carbon, that's 2 electrons. Well, it only needs 2 more, so it's sharing an electron with that hydrogen. Okay, and it's sharing an electron. It's got a double bond there with that carbon, so it doesn't need any more electrons, right? So you have a hole right there. That's what unsaturated means. It means that this carbon chain is not saturated with all the hydrogens it could possibly hold because it has these double bonds. Okay? So the carbons are not fully bonded with hydrogens. That's why it's called unsaturated. Well, take a look over here at the space fill model for this molecule. This is literally showing me what the molecule would look like in a space, okay? And notice that where there are these double bonds right there, it causes that fatty acid to bend or kink, okay? So double bonds result in kinky fatty acids. Well, these kinks prohibit those molecules from stacking on each other nicely. So when you stack a whole bunch of those molecules together, they don't fit together. And that's what causes those substances to be liquid at room temperature. Those kinky fatty acids. Okay? So take a look now at the saturated fat. Right here. Those were the ones that were from animals or coconuts. Okay, tropical oils are saturated. Okay, there are no double bonds in the fatty acids of saturated fats. Notice that all the carbons right here are bonded with single bonds to either another carbon or a hydrogen. So there are no double bonds. The carbons are saturated with hydrogens. Well, since there's no double bonds, it causes this molecule's fatty acids to be straight. You have straight chain fatty acids. Well, if you take this molecule and you stack it on top of other molecules just like it, they stack very nicely together because they're straight. It's like stacking books up, which causes this substance to be solid at room temperature. So that is the difference between saturated and unsaturated fats. Okay, If you'll remember, let's go back to our structure right here. If you remember, the molecule of fat and oil was a glycerol and 3 fatty acids. This structure, as it is hydrophobic. There are no polar regions on this molecule. This molecule is nonpolar. It has no polarity. Okay? We're going to move now into a group of lipids called phospholipids. Okay? And they look just like this, except this third fatty acid right there is replaced with a phosphate group. So let's take a look at that. A phospholipid has a structure similar to fat, except the third fatty acid is replaced with a phosphate group. So it looks like this. You have a glycerol, you have 2 fatty acids, and right here is a phosphate group. Without the phosphate group, remember it was nonpolar. Well, the phosphate is a functional group. Do you guys remember when we said you add a functional group, you can change the polarity of the molecule? Well, this phosphate group right there has a charge associated with it. Okay? That group right there is polar. And what happens is that phosphate hates those fatty acids. I mean, literally hates them. Okay? Because they're nonpolar. So this phosphate group, and I don't know if I can get the pen to work. Nope. This phosphate group literally stretches all the way to the other side so that here it is right here. Here's the glycerol, here's the 2 fatty acids that were right there, and the phosphate group stretches all the way to the other side to get away from the phosphates. It's trying to put as much distance as possible between itself and the fatty acids. So it literally looks like this. There's the phosphate group, there's the glycerol, and there's the fatty acids. The phosphate is polar. The fatty acids are nonpolar. So when you look at this molecule, we refer to the phosphate as the head of the molecule, and it's polar. And we refer to the fatty acids as the tails of the molecule, and they are nonpolar. Okay? So the tails are going to be hydrophobic, and the head is going to be hydrophilic. Yes. So now this molecule has some polar regions. Very important that you understand the nature of this molecule, because this molecule is extremely important to the rest of the course. We got a whole chapter that deals with this molecule. Okay? Because these phospholipids make up the covering that goes over the cells of your body, the plasma membrane over your cells. Every cell has a covering called a plasma membrane, and it's made primarily of these phospholipid molecules. So I want you to think about that for a moment. We've got a molecule that has a head that likes water and tails that hate water. Right? And it's going to cover your cells. But your cells, both inside and out, are 70 to 90% water. And those tails hate water. So what happens with these phospholipids is when they're in the presence of water, the tails try to get away from the water. So they're going to form a double layer around your cell to insulate the tails from the water. Okay? They form a double layer. We call it the phospholipid bilayer. Bi means 2. So let's take a look at what that would look like if these phospholipids formed a double layer. It looks like this. With this area right here being inside the cell, this area out here would be outside the cell, okay? So this would be the plasma membrane. This is the covering over every cell in your body. You see, here are the heads of one layer and the tails. So this makes up one layer. And here are the heads of the other layer, and the tails. So you've got a double layer so that the tails never are exposed to the water. Does that make sense? So this area in here is hydrophobic, and this area out here is philic, and this is, philic. This is hydrophilic, that's water. I know this is so hard to visualize, guys. It really is. I want you to think of it like a baseball. And the cell membrane is the covering, that leather covering over the baseball. Okay, it's made of this. So let's look at it three-dimensionally like a baseball. Okay, let's pretend that you're inside that cell. You're sitting right there, and you pull the cover down over you. If you're sitting right there and the cell is put back together, what of those phospholipids are you seeing? Only the heads, right? So let's say you get out and you put the cell back together. If you're standing outside the cell and you're looking at it, what of those phospholipids do you see? The hydrophilic heads. So you never see the tails. They're inside the membrane. Okay, does everybody visualize that now? Very important, because we have a whole chapter that deals with how molecules go in and out of this cell, how they get through those phospholipids, because remember, that's hydrophilic, hydrophobic, hydrophobic, hydrophilic. Okay, so a molecule would have to go through all of that to get to the inside or to get to the outside. Okay? Steroids, not going to spend a whole lot of time on steroids. These are lipids that have 4 fused carbon rings. Let me show you what those look like. This is a carbon ring right there. This is a carbon ring. That's a carbon ring. Wherever there is a joint there would be an understood carbon. So that's got 5 carbons. That's got 6 carbons, etc. So all of these, they're fused together. All of these are lipids. And examples would be the testosterone here, the estrogen there, the cholesterol. Those are all steroids. Okay, let's continue on with waxes. Most of you know what a wax is, or you know when you see it. It's molecularly, it's a long-chain fatty acid with an alcohol. A long chain, there's your fatty acid and there's your alcohol right there. With regard to the characteristics of waxes, they are solid at room temperature. They are waterproof. They're hydrophobic, okay, non-polar. They serve protective functions in animals and plants. Some of you may not have been aware that plants have wax. Where would you find wax on a plant? On the leaf, yeah. Some leaves are waxier than others. They're there for protection and to help the leaf from drying, keep it from drying out or desiccating. A great example of a waxy leaf would be a magnolia. Ever seen a magnolia leaf? It's very, very waxy. There's some plants with wax. Where do you have wax? In your ears. Is that not gross? Yes. Wax is waterproof. It protects your ears. It's sticky. It keeps things from getting in your ears. It doesn't smell good. It doesn't taste good. So it helps to protect your ears. Okay? Okay, let's go into proteins. Okay. Proteins, lots of different structures of proteins result in a lot of different functions of those molecules. For most of you, when I say the word protein, you guys think of a juicy steak or a hamburger or something like that, right? That's because that's where you get a lot of your dietary protein, okay? But keep in mind, we're talking about organic molecules here. And you are made of the same things, the same molecules as the food that you eat, literally. So there are lots of different places that we find proteins in your body, though, other than muscles. And that's what you're eating of another animal when you have a steak. So let's talk about those other proteins. Not that the muscles are not important, but proteins are so much more than muscle. Sounds like a commercial, doesn't it? Proteins do so many more things in your body than just form your muscles. The collagen that's holding you together, literally, is made out of a protein. It's for support. The enzymes that control your metabolism, those are proteins. We have a whole lot of chapters that deal with reactions and enzymes. They're catalysts. The hemoglobin that's found in your red blood cell that binds to the oxygen and carries it through your bloodstream, that molecule is a protein molecule. The antibodies that are part of your immune system that defend your body against foreign invaders, those are proteins. Insulin, the hormone that helps control your blood sugar, that is a protein. And last but certainly not least are the muscle fibers that make up your muscles. These are also proteins. So notice that there are lots and lots of different molecules that are proteins. So as we talk about proteins, I want you to think about more than just muscles, okay? The monomer for a protein is an amino acid. Amino acids, all amino acids, have 4 parts. They all have a carbon bonded to a single hydrogen. It's right there. They all have what's called an amine group. It's right there. Amine groups contain nitrogen. They all have an acid. It's called a carboxyl right there. And amine and carboxyl are functional groups. Notice that my hydrocarbon chain is now a carbon and a hydrogen, okay? And all of the amino acids have something in this area right here that for right now we're just going to call the rest of the molecule, okay? So all amino acids have 4 parts. Carbon with a bonded to a hydrogen, an amine group, a carboxyl group, and something right there. Okay? There are 20 different amino acids. So obviously they all have those parts. So what's going to make one amino acid different from another amino acid is what's right there in the R position. Some call this a side chain. And you may hear me, refer to the R group as a side chain. Okay, so basically there's 20 different side chains, right? There's 20 different combinations of atoms that would go right here in that space. So the unique properties of each amino acid depends on what's there. So let's take a look at the amino acids for a moment. If you're a bodybuilder or a sports person and you're drinking protein shakes, you may recognize some of these amino acids, okay? Glycine, alanine, valine, leucine, tryptophan, that's the one in turkey, proline, some of these are really common that we've heard of. Notice that of the 20 amino acids, all of them have this, it's kind of a violet color right here, a blue. They all have that in common. That is your carbon with the hydrogen, your amine group, and your carboxyl. So they're all highlighted the same, indicating that they're the same for all 20 of these molecules. Okay? The R group is highlighted for this area, for this group of amino acids. The R group is highlighted yellow. For this group of amino acids, the R group is green. And for these, they're pink and blue. Those are the R groups of the side chains. Notice there are 20 different side chains. They are grouped based on the properties of the R group, or the side chain. All of these side chains are hydrophobic. All of these side chains are hydrophilic, and these side chains are charged. They're acidic or basic. Okay? So the side chain or the R group has properties associated with it. Is everybody with me? Okay. So when 2 amino acids bond together, we call, what would be the reaction that bonds 2 amino acids together? Dehydration or condensation synthesis, yes. When 2 amino acids bond together, the bond that forms between them is called a peptide bond. So what you have here is that is an amino acid bonded to another amino acid, so that would be a polymer. Okay, here's an amino acid that's being bonded to this polymer. And notice, just as I mentioned earlier, this monomer has a hydrogen right there and an OH right there. So this is condensation synthesis. We take the H from the monomer, the OH from here, water comes out, and now we bonded that amino acid to the polymer. Okay? So this would be a polymer of amino acids. There's the side chains to those amino acids right there. As a matter of fact, we're going to call lots of amino acids bonded together, we're going to call that a polypeptide. Just like we called lots of monosaccharides bonded together a polysaccharide, we're going to call lots of amino acids bonded together a polypeptide. As a matter of fact, right now in your notes, I want you to write down peptide equals protein. The word peptide is associated with proteins. So anytime you hear that word, you know it has something to do with a protein. Okay? Okay, so that's a polypeptide. And a protein would be one or more of those polypeptides. Okay, so let's talk for just a moment. Protein shape is critical. You're not going to have to write any notes on this particular slide. I just want you to look at the shapes of some of these different proteins. Okay, that is collagen. That's an antibody. Those are enzymes, and this is DNA polymerase that's involved in synthesizing DNA. I want you to look at the variety of shapes that proteins come in. Protein shape is critical. It's the most important thing. It's how a protein works. Have you ever tried to put together a jigsaw puzzle? Do y'all know what jigsaw puzzles are? And you know that you have to match the shape of the pieces together to make the picture come out right? Okay, so shape is critical. Proteins work like jigsaw puzzle pieces. They literally fit into other molecules, which causes things to happen. Okay, so if the shape of the protein is not right, then it can't do its job. It can't work. So the shape of the protein is very specific. It's not an accident at all. It's very deliberate, and it's very important in the function of the protein. Okay, so let's talk for a moment about how does a protein get that shape? What determines the shape of that protein? Okay. First of all, I want you to, I wish I could use the symposium. I don't know how to use this one, though. I'm sorry. I want you to think about the language, our English language. How many letters are there in the English language? How many letters? 26. Okay, how many words can you make with 26 letters? A lot. That's exactly right. Well, I want you to stop for a minute and think about what makes a word a word. If I write the word ate, A-T-E, what makes that word ate? What makes you recognize that as the word ate? Do what? The letters in the word, right? Well, what if I mix them up and I change it to E-A-T? Is that still the same word? No. So we recognize a word because of the letters that are there in the right order. They have to be in the right order, right? Well, what happens if I add a letter? Let's say I take A-T-E and I put an R in front of it. Is it still the same word? Not at all. I added a letter and it changed the word totally. What if I have the word A-T-E and I take the A off? I'm sorry, back up. What if I have the word A-T-E and I take the E off? It changed the word, didn't it? So a word is a word because of the letters in the word and the order or the sequence that they're in. If I change the sequence, it's not the same word. If I add a letter, it's not the same word. And if I take a letter away, it's not the same word, right? I want you to start thinking of proteins the exact same way, okay? There are 20 different amino acids. And what defines a protein? Just like a word, what defines a protein is the amino acids that are in the protein, and the exact order that they're in. Okay? Just like the word, this is so important. The sequence of amino acids is so important that we're going to call the sequence the primary structure of that protein. So the primary structure of the protein is the sequence of amino acids in that protein. Okay? So just like our word, guys, if you change the sequence of amino acids, it's not the same protein. If you add an amino acid, it's not the same protein. If you take an amino acid out, it's not the same protein. Okay? Because what you have here in the illustration, look at this. If each one of these is an amino acid, and you've got these R groups that are hanging off the side, right? And you saw in the previous slide that those R groups have properties associated with them. Well, some of the R groups are hydrophobic, some are hydrophilic. So you've got this long chain of amino acids. The ones that are hydrophobic are going to want to get away from the water. Right? So they're going to try to pull in and use the other amino acids to protect them from the water. The ones that are hydrophilic are going to love the water. Well, there are even some R groups, y'all, that like each other, and they bond together if they're close to each other. So if I have an amino acid with an R group here, and let's say I have 20 other amino acids in between, and I have another amino acid here with an R group that they like each other, the two on the end like each other, they're going to try to get to each other. So what does that do to that long strand of amino acids? It's going to cause it to fold or pleat. Okay? So when you get a protein made, when you get all of these amino acids bonded together, those R groups start interacting with each other. They start forming these relationships. They start interacting with the environment. Okay? Those interactions are called the secondary structure of the protein. That's what happens when all of those R groups start to interact and you get folding, like this right here. You get pleating, where the R groups try to come together and bond. Sometimes you even get these helices, coils. You get these coils. And y'all, every single interaction is necessary. If anything about it changes, the protein changes. It's not going to work because all of these interactions ultimately lead to the third level structure of a protein, which we call the tertiary structure. And that's the shape of that protein. So let's recap that for a minute, guys. This is really important, extremely important. This theme carries over to about 6 chapters, okay? This structure right there that you see, that protein molecule, is shaped that way for a purpose. It has a job to do that is a function of that shape, okay? Every curl, every angle, every straight part of the molecule is necessary, okay? That shape is a result of all of those interactions between the R groups and the R groups with the environment. Those interactions are defined by where they are in the chain. Where they are in the chain determines the relationships which determine the shape. Does that make sense? Some proteins are huge, like hemoglobin is an example, and it takes more than one polypeptide to make the protein. When a protein involves more than one chain of amino acids, more than one polypeptide, we say that it has a 4th level structure. It's called the quaternary structure. That's the overall protein structure that results from the combination of 2 or more polypeptides that come together. And hemoglobin is a good example of that. Hemoglobin is similar to this illustration right here. It has 4 subunits. And the 4 subunits come together to form the hemoglobin molecule, and the oxygen literally sits in the middle of the hemoglobin on your red blood cell. Right there. As a matter of fact, guys, to tell you how important the exact sequence of these amino acids actually are, there are some individuals whose DNA actually codes for one amino acid wrong in their hemoglobin protein. And remember, hemoglobin is 4 different polypeptides, so we're talking about hundreds and hundreds of amino acids. And these individuals have one that's wrong. One amino acid out of all of those hundreds is actually wrong. And those individuals, the oxygen will not bind to the hemoglobin because the property of the protein has changed, and those people have sickle cell anemia. One amino acid wrong. Very serious. So shape is critical. Proteins can actually lose their shape. The shape is dependent on the sequence and the physical or chemical environment. So if you change either of those, you could change the shape of the protein. Things like temperature, pH, etc. are very important to protein structure. This is why homeostasis is so important. If you get a high temperature, you start changing the environment. These proteins are very sensitive to that, and they literally will change their shape. And if they lose their shape, they're not going to work. Okay, so if we're talking about enzymes, you're talking about your metabolism and death. Okay, we call the change in protein shape denaturation. It's denatured. It loses its shape, and when it loses its shape, it's not going to work. It's not going to do its job. Okay, let's go to our 4th group of organic molecules called nucleic acids. Some of you may be familiar with some of these. They have 2 functions in general. There's a group of nucleic acids that are used to store and transmit genetic information. There's also a nucleic acid that is involved in energy transfer in living systems from one molecule to another. We're going to talk about those today. There are 3 types. We're going to use these are the 3 examples of nucleic acids that we're going to talk about. The first 2 are to store and transmit genetic information, and that is DNA, or deoxyribonucleic acid, and RNA, ribonucleic acid. So those 2 are involved in the storage and transmission of genetic information from one generation to the next. The third nucleic acid is involved in energy transfer from one molecule to another, and that is ATP, or adenosine triphosphate. The monomer of a nucleic acid is called a nucleotide. These are the building blocks of nucleic acids. They bond together to form the nucleic acid. Every nucleotide has 3 components, 3 parts. It's going to have a nitrogen base, which is this area right there. It's going to have a phosphate group, which is that area right there for this example. And it's going to have a 5-carbon sugar, or a pentose sugar, right there. 3 components: a nitrogen base, a phosphate group, and a pentose sugar. There are actually 5 different types of nucleotides. You see them right here, they all have the same structure with regard to the phosphate. There are 2 different sugars involved, but they're very, very similar. Where these 5 nucleotides differ are the nitrogen bases right here in this area. And they are called adenine, guanine, cytosine, thymine, and uracil. Adenine, guanine, cytosine, thymine, and uracil. DNA contains the first four. DNA would have adenine, guanine, cytosine, and thymine. Notice that thymine is highlighted in yellow. RNA contains adenine, guanine, cytosine, and uracil. Adenine, guanine, cytosine, and uracil. Notice that RNA and DNA have basically the same nucleotides, except for the thymine in DNA is replaced with uracil in RNA. The nucleotides join together through, what is that reaction? Dehydration synthesis, yes. Condensation synthesis. The nucleotides are going to join together to form a linear strand or polymer of nucleotides. I want you to take a look at this illustration right here. Here is my nucleotide right there. There's my base, my pentose sugar, and my phosphate. And this is how the nucleotides join together. The easiest way, when you see a diagram like this, the easiest way to identify a single nucleotide, to me, the easiest way, is to identify the sugar first. Okay? We're talking about pentose sugars. What shape does a pentose sugar take? It takes the shape of a pentagon. There's 5 carbons, yes. So if I look in this molecule, this strand, if I find the pentagon right there, that tells me that's the sugar. Well, we know connected to the sugar in the nucleotide is a phosphate and a base. So that right there is one nucleotide. That's a sugar, a phosphate, and a base of a nucleotide, a single nucleotide. Okay, the phosphate of one nucleotide bonds to the sugar of the next, and these are covalent bonds. So if this is the sugar of the next nucleotide, there's the nitrogen base and there's the phosphate. So there's my second nucleotide in this strand. That phosphate is bonded to the sugar of the next nucleotide. So there's my next nucleotide right there. So this is a polymer of individual nucleotides, the monomer, bonded together to form a single strand of nucleotides right there. Okay? Let's talk about DNA and RNA and some of the differences between them. Again, the sugars and the phosphates that bond together to form the polymer are referred to as the backbone of the molecule. That's where they bond to form the single strand of nucleotides. And RNA is a single strand. This is an actual example of what RNA molecularly would look like. There's my sugar, there's my phosphate, there's uracil, that nitrogen base. There's the sugar, there's the phosphate, that's adenine. So that's the second nucleotide there. So that would be the beginning of an RNA molecule. It is single-stranded. RNA is a single strand of nucleotides bonded together, A's and T's, no, not T, A's and G's and C's and uracil, yes. Okay? DNA is a double-stranded molecule, A's and T's and C's and G's, okay? You probably think of DNA like this right here, to your left, as a double helix molecule. What I'd like to do for illustration is to take that molecule, and if you're thinking of that as like, let's say, a spiral staircase, okay? The gray portion right here would be the handrails of the staircase, and the colored portions would be the steps, or the rungs of the staircase, right? So if I take that spiral staircase, I'm going to unwind it and lay it flat so that it looks like a ladder. Okay, so let's take this and go over here, and this is going to be my ladder now. The handrails, the gray portion, would be the blue and yellow parts of this illustration. So that would be one of the handrails, and here would be the other handrail. And the colored steps would be in the center right here. So now let's take our ladder and look at the components, and let me show you where the nucleotides are. First of all, this ladder is actually 2 different strands of nucleotides hydrogen bonded together. These broken dotted lines right there represent hydrogen bonds, and we learned what that was in Chapter 2, that was an attraction between a positively hydrogen atom of one molecule and the negative area of another molecule. And we also learned that those are weak bonds unless you have lots and lots of them, and then they're really strong bonds. Okay, so these bonds are pretty strong right there. There's lots of them. In all of your DNA, approximately 3 billion of them. Okay, so those are hydrogen bonds. Let's look on either side of the hydrogen bond there. This would be a single strand of nucleotides bonded together, and this is a single strand. So here we go. Here's the sugar, the base, and the phosphate of one nucleotide. That phosphate is bonded to the sugar of the next, the base, and the phosphate, and so forth and so on. We have the same thing on the other side. We have a sugar, a base, and a phosphate. Some of you may be saying, well, they don't look the same, though. Well, they're going in opposite directions. We call that anti-parallel. We'll talk more about that later. Look at the sugars. The easiest way to see that they're going in opposite directions is to look at the sugar molecule on either strand. The sugar is a pentagon. It's a pentose sugar. On this side of the molecule, the sugars are pointing upward. On this side of the molecule, the sugars are pointing downward. Okay, so they're going in opposite directions. But the two strands are held together by hydrogen bonds. They twist to form this double helix that you recognize as that DNA molecule. When they bond together with hydrogen bonds, adenine always bonds with thymine, and cytosine always bonds with guanine. We call that complementary base pairing. So A is a complement to T, C is a complement to G. Okay, let's move on to our next nucleic acid. ATP, a very special nucleotide. It's called adenosine triphosphate. ATP. It is a nucleotide. It is made of an adenine, ribose, which is the 5-carbon sugar, and instead of having one phosphate, it has 3 phosphates. That's why it's called triphosphate. So it's adenosine triphosphate. Adenine, ribose, and 3 phosphates. Okay? Guys, ATP is an exception. You might want to put exception. It does not polymerize. What does that mean? That means it's not going to do what? It's not going to bond together to another ATP. Yeah. Okay? When you had DNA and RNA, you had the nucleotides that bonded to other nucleotides that bonded to other nucleotides. Yes. ATP does not do that. What you're looking at right there is the total molecule. It's not going to bond to another ATP. Okay, it does not polymerize. It is a very high energy molecule. As a matter of fact, the energy in the molecule, and pay close attention, the energy in the molecule is right there. That third, the bond right there between the third and the second phosphate group has a lot of energy. We call this molecule the energy currency of the cell. Why do we call it the energy currency of the cell? It's because cells literally spend it. Okay? When a cell needs energy, that cell is going to break this bond and use the energy that's released from that bond. Okay? Every cell in your body, every function in your body that needs energy gets its energy from this molecule right here. Okay? The hydrolysis of that third phosphate bond yields the energy that the cell needs. Okay, let's look at that for just a minute. If I take that phosphate off right there, if that phosphate is not there anymore, what do you have left? You have adenine, ribose, and 2 phosphates, right? What's a prefix that means 2? Di. or we'll use di in this case, okay? So if we take that phosphate right there off, we would have a loose phosphate over by itself. We would have ADP, adenosine diphosphate. We only have 2 of them. And we would have energy released, right? So we have ADP, a phosphate, and energy. That energy is where your cells get all the energy to do their work. Very important reaction. Okay, make sure that you understand this right here. This is your ATP molecule. When that bond between the second and the third phosphate is broken right there, energy is released. Okay? So you have ADP plus P. Does anybody have any clue what happens to that? Yes. Does anybody remember the mitochondria? What is it? The powerhouse of the cell. Yes, everybody remembers the mitochondria as the powerhouse. They have no idea what that means, but we remember it's the powerhouse. Well, in the mitochondria, the reason it's called the powerhouse is because in the mitochondria, your mitochondria actually takes the glucose from the food that you eat, breaks down that molecule, and uses the energy to rebond that phosphate back to ADP. So this part of the cycle, energy input, happens in the mitochondria. That's going to be the energy from the food that you eat, the glucose, it's broken down to, and your mitochondria is going to use that energy to make ATP.
Is that ribose on the mitochondria? Is that ribose on
Ribose, it's going to use this to, we got a whole chapter on that, the next chapter, okay? So the mitochondria builds ATP for us. Your cell uses it. The mitochondria builds it. Your cell's going to use it. The mitochondria builds it. Your cell's going to use it, et cetera, et cetera. So this is the reaction. It's a reversible reaction. ATP is going to break down into ADP plus P plus energy, and it's going to go back the other way. So cells use it, mitochondria builds it. Cells use it, mitochondria builds it. Okay? Also, I'd like for you to take a look at this illustration. I stole this from a book somewhere. It may have been your book. I can't even remember. It's not all inclusive, but I liked the presentation. They organized the information well. Guys, you're going to have to really get organized to study for this exam. I liked the way they presented this.